Thank you, Marcus. Um, so, uh, delighted to be here and to give a presentation on artificial metallo-nucleases. And I'd just say it's an extremely hard act to follow Tom, and particularly when he's got a Lifetime Achievement Award. So I'm doomed from the start. But anyway, just like to start by saying it works. Oh yeah, so I've just got a couple of words to say about Tom, and I wanted to share. I've had the good fortune of coordinating two large-scale Marie Curie awards. One was called Click Gene, and the other one was called Nature ETN. And the idea here was that we would train new scientists in the area of nucleic acid chemistry. And together with Tom and leading nucleic acid chemists in Europe, Thomas Carell, Mikhail Hocek, Afaf El-Sagir, we developed these two networks. And the kind of applications for both were, as I said, to train new scientists in nucleic acid chemistry, but also to develop new ways of applying click chemistry to gene editing tools. And I must say, throughout, Tom has been an incredible driving force within both of these networks in terms of his encouragement, willingness to support students, and for his openness. I think, to be honest, we were focused at the time ClickGene was funded was 2014, and we were very focused on DNA damage. And what it allowed me in terms of the collaboration was to learn a lot more about gene editing tools and this whole area. So as I said at the beginning, I owe a lot to my own career, I think, to interactions with Tom and the other nucleic acid chemists here. And the other thing to mention is that with Tom, he's been very helpful in terms of organizing meetings. So through this, for example, the Oxford Oligo meeting, and also other training networks. We've had the chance to bring some of these students here, and they're all doing extremely well at this point. A lot of them are working in industry, several are in academia, and so it's kind of testament to training the next generation. And I will say in this photo here, we still don't know who this individual is, but maybe a good photo to get into. But there's a reward if we ever find out. But yeah, so these are the networks that we're part of, and absolute pleasure to work with them. The other thing to mention, of course, is that as a byproduct of these collaborations, we have been publishing extensively in the areas of triplex forming oligonucleotides, new DNA damaging systems, and also in developing hybrid gene editing systems. And as I said, the impact of this work is a byproduct of the collaboration. And as Afaf said, the focus is really not on where we can publish, but just the curiosity. And our most recent work was featured on the cover of RSC Medicinal Chemistry. And we have several other ongoing projects, which I'm looking forward to continuing to work with Tom. Okay, so on to my own talk then. So what I want to do today is focus on artificial metallo-nucleases and our ability then to use click chemistry to develop these types of systems. We can then incorporate these into oligonucleotides, making them catalytic as kind of artificial gene editing systems. And I'll talk briefly about this in part 3, but my colleague, Dr. Brina McGorman, will speak in a bit more detail during her talk about that and recent developments that she's been leading. And in the final part, I want to talk about this infrastructure fund. Okay, excuse me. The two main mechanisms by which we can cleave DNA is through hydrolysis. And here you can see that for example, the DNase 1 cleaves DNA, and we end up with an intact 3 and 5' ends that are amenable to religation and repair. On the opposite side, you can use oxidative mechanisms. So this is where you have a radical species that attacks, for example, deoxyribose or the base, and this produces a wider variety of breakdown products that are more difficult for the cell to repair and more energetically expensive. And so we're interested in this area, and these can be generated from metallo-nucleases with metal-centered radicals or oxo species. And so some of the inspiration here is taken from metallo-bleomycin, and this has a bithiazole unit that can intercalate DNA, and then in a neighboring region, you combine with a metal ion to produce ROS or reactive oxygen species that produce oxidative damage. I think one of the interesting things about these systems is that they have the ability to cleave DNA using different ROS mechanisms. It's not exactly like Fenton chemistry, but it's somewhat similar in that the metal center has an oxo group bound. So we're not always producing diffusible radicals. But one of the key things here is that depending on the shape, charge, and size of the group of the metal ion, you can cleave DNA at different positions. And so these are kind of tunable type systems. And in general, in the field, what we can say is that there's not a huge amount known in terms of the binding ability or the sequence specificity of these compounds. But the limited knowledge that we have, we can see, for example, like cisplatin interacts or crosslinks at guanine-guanine groups here to prevent DNA replication. If we have this TA-TA step, we can Nature ETN grant, we've contributed or expanded upon these limited systems. And what we found together with Christine Carden and our PhD student Taylor Prioto-Otoya, is that we've characterized the first major groove binding ruthenium compound, this Phi compound. And this is a cover article in Angevin Chemie several years ago. And we also were able to detect or use the same compound for double mismatch detection. And so this kind of area is in its infancy, but what we can see is that these metal complexes have very specific interactions, whether it happens to be a TA, AT, mismatches, and we can design them to bind in certain ways with nucleic acids. So what we do in our group is we build artificial metallo-nucleases. These often have multiple metal centers, and what we're interested in doing then is to target these using click chemistry. So this is where our collaboration with Tom comes in. We can design, for example, triplex forming oligonucleotides. We can then click these artificial metallo-nucleases and generate these hybrid systems. And the overall goal then is that we can drive these interactions sequence specifically, and the type of editing here is different to state-of-the-art gene editing tools like CRISPR or zinc finger nucleases. And so I'm not going to talk extensively about this. Dr. McGorman will highlight a lot of our recent work in this area, but I'm going to focus on our kind of development of artificial metallo-nucleases. And so we've had some success in this area. We start off with the standard artificial metallo-nuclease Sigmund's reagent discovered in the 70s. We've been able to improve the stability of this by caging it and putting DNA intercalators into the system. we've been able to, together with Tom, look at these dinuclear compounds which have interesting Z-like properties. And these occur at the TA step, which is really quite interesting given the recent structural evidence for ruthenium compounds. And what we've had success most recently with is these tri-click systems. And what we've been able to do is use click chemistry to build artificial metallo-nucleases where the N group from the triazole serves as a chelating or a donor group as part of this kind of scaffold. And so I want to talk about the development of these systems in the talk. Of course, directing these groups is not new. There's some, going back, I think, historically, some really interesting work in terms of using minor groove binders like distamycin, hooks. BQQ, for example, targets triplexes. Spermine enhances the electrostatic interactions. And then more recently, Cowan has shown that you can target the ATCON, which is a cleaving group, to acridine. What none of these groups have done is use click chemistry to build kind of directed systems. So we've been able to do that using either small molecules or triplex forming oligonucleotides. So there's a lot of advantages in the modularity of that approach. So our kind of, we serendipitously discovered these kind of click-based artificial metallo-nucleases, and it started from work with Natasha McStay. And Natasha made these mesethylene, these C3 symmetric opioid compounds, and she found that when you protonate the piperidine ring, they serve as very good condensing agents. So this is AFM imaging. And there's obviously disadvantages in terms of using opiates as condensing agents. So we wondered if you could simplify this system. And for example, could we take this tri-click, so this triazide, again around the mesethylene core, and then click on a range of primary, secondary, and tertiary aliphatic and aromatic amines, with the idea that being that they could become protonated and then serve as condensing agents. And it turns out they were no good. And I said then to Natasha, look, to try and maybe rescue the project, could we put in first row transition metals to see would any of them have cooperative effects? And again, very limited results. However, with copper and TC1, where we have the NN group here in this chelate binding mode, we found that it had excellent DNA cleavage activity. So this is PUC19, where it's cleaving to the open circular form in the absence of any reductant. The binding is extremely high in the presence of copper, and Natasha also characterized the DNA damaging properties of the compound. So really interesting discovery then, but completely by accident. Dr. Alex Gibney, or Alex at the time, who joined the group and wanted to explore this chemical space to find out, well, does it just need to be a primary amine attached at this point, or are other groups amenable? And so one of the first molecules he made was this. This is an ethinyl thiophene, and this led to the discovery of TC-thio. And this molecule, we'd expect the nitrogen group to bind with copper-2, but upon reduction in cellular environments to copper-1, the sulfur would become active and chelate. And so here's the crystal structure of the compound. We found that it binds 3 copper ions very efficiently, and then at least computationally, it shows that the sulfur and copper interact upon reduction as anticipated. We have high throughput screening assays. Some of these are relatively simple, like ethidium bromide displacement or thiazole orange displacement. The binding is dependent on copper. We can simply displace the bound fluorophore. And then we can also do a little bit of geography, where if we look at either a major groove binding fluorophore or a minor groove, we can see that, for example, Hooks is displaced much more readily. In terms of static interactions computationally, we found that 2 of the molecules are sitting in the minor groove, or 2 of the arms effectively are sitting in the minor groove, with a third one swinging out or not engaged. And the kind of major driving force, at least using the static model, is electrostatics or Coulombic interactions. But one of the assays we've developed has been inspired from Tom's work on molecular beacons. And what we've been able to do is use these hairpins. This is the Dickerson-Drew sequence. And if we use an unlabeled oligo, we can apply something like circular dichrosum spectroscopy to look at conformational changes. If you introduce a fluorophore like Psi5, we can then use microscale thermophoresis to give us direct binding measurements. So this is the same construct, but just with a fluorophore. And then if we introduce a quencher, we can look at thermal melting properties. And as a result of this, we can run these on a light cycler instrument. So we can effectively do a range of concentrations very quickly, and we can extract this data and plot the direct KD value, or find the KD value from the Bard equation. And one of the key assays we've developed, or we've collaborated with Professor Frederick Westerlund, is this single molecule imaging assay that his lab has pioneered. Basically, you can take primary human blood cells, extract these, expose them to your DNA damaging agent of choice, and then afterwards you can look at the extracted DNA, expose specific base excision repair enzymes, or a cocktail of them, and then introduce a fluorescently modified DNTP. This then incorporates a label, like a foci, that can be measured as a function of a general stain like yo-yo-1. So we can measure the number of damage sites, but also the assay can be layered, whereby you might use a range of BERs or single BER enzymes, or you could introduce scavengers. And what we were able to do in this assay is identify the activity was similar to the clinically approved temozolomide, and that the activity when we added something like Tyron returned to baseline, which is a superoxide scavenger. So really nice work. And we ended up publishing this as a cover article in Angevante Chemie, together with Frederick and some other people across Europe. So very recently, we've had a paper accepted in Nature Communications, where we asked another relatively simple question, which is, well, we know what TC1 and TC-thio are and how they function, but what happens if we expand the chemical space? So if we use something like alcohols, acids, amides, and then some of these aromatic amine groups like pyridine, pyrimidine, benzothiazole. And what we found in this system, so this work again was led by Alex and a more recent PhD student, Eva Delahunt. And what we found was that with these nitrogen heteroatomic systems, the TC-pyridine and TC-pyrimidine, for example, these gave exclusively trinuclear compounds, and they also had the highest DNA binding affinity from the overall system. So we pursued TC-pyridine in more detail, and again, applying some of these similar techniques using hairpins, we were able to look at the binding towards the Dickerson-Drew sequence, characterize the binding using thermal melting or fluorescence melting. And then together with Frederick Westerlund, we could characterize the compaction properties and interaction with single molecule DNA. And we then applied more sophisticated molecular dynamics. And what we found again was that the molecule binds in the minor groove. You've got two sites, or two of the arms effectively are engaged, with a third one swinging open. And again, the activity is driven coulombically rather than through van der Waals forces. So we're building a kind of picture now, of more detailed picture of how this molecule is binding with DNA. And work from Eva then, which was really quite interesting to see, was that we developed a whole range of these hairpin sequences. This is the Dickerson-Drew. We can substitute, for example, cytosine with methyl cytosines to effectively block the major groove and direct the compound to the minor groove. We were able to look at Tata-rich sequences, long runs of GCs, interrupted runs, and then mismatch sequences, single and double mismatch sequences. And what we found here was that the molecule, the copper TC-pyridine, has very high thermal stabilization for continuous runs of GC sequences. If we interrupt this with AT, we still have significant stabilization. And coupled then with the methyl cytosine derivative, which is blocking the major groove, we can again see the activity in the minor groove is driving this thermodynamic response. And interestingly, with these mismatches, we found no stabilization occurring, so they're not displacing any of these mismatch bases. We then collaborated with the National Cancer Institute in the US and found that the ligands alone have interesting activity. In general, we found that certain cell lines, in particular triple negative breast cancer cell lines, were sensitive to the TC-thio ligand. And this was borne out then in a smaller assay where you can see its IC50 value is actually much greater than what we found for the earlier TC-thio that was reported. And then in a final set of experiments, these are kind of functional assays, and effectively we can monitor the amount of copper being uptaken in certain cell lines. And so copper is being taken up by the compound. The cell cycle analysis indicates that the TC pyridine compound is damaging DNA, and we can identify this through a particular checkpoint. It's very active in cleaving DNA and in particular producing double-strand breaks. And then when we look at functional assays with bacterium, what we find is that the molecule disperses the genetic contents and gives high-resolution cleaving activity similarly to nitrofurantoin. And so you can see here that the average length, which is measured in microns, is much shorter compared to obviously the untreated DNA here. And as I said, that work has just appeared last month in Nature Communications. Where we're going to go with this is Eva's kind of taken up the mantle for the triclic compounds. And what we're interested in looking at now is derivatizing these systems further. So up to now, we just have two donors, the triazole and the donor group here. But if we introduce, say, a functionalization like picolinic acid, the idea here could be that we would form a more stable variant where you've got three donors, or for example, these kind of higher order denticity structures that could wrap around the compound. So Eva has a poster on this research at this conference, so you might be interested to learn a little bit more about that work. One of the questions now is how do we direct these to specific sequences? And so Oliver Gold, who's here as well and has a poster on the work, has very recently completed a study where I think the breakthrough here really is in using mesotol. So what we can do with this is we can introduce our bromide handles here before azidation, and then we can put on two of these pyridine groups, similar to the TC-thio, obviously, or TC pyridine. And this leaves the alcohol here, which can then be later functionalized. And in this case, we just simply looked at the conjugation of an acridine group. But in general, this could be, we could attach, for example, an oligo, a triplex forming oligonucleotide, or another directing group. And so this leads to a click chemistry type hybrid. So I think this is going to open the door for new types of hybrids based on these click chemistry based AMNs. And the interesting thing about this work is that because only two of the arms are engaged in the minor groove, this leaves a third site, which we've effectively repurposed for this directing group. In this case, is the acridine donor. And the endpoint here is that when we pre-expose DNA to actinomycin D, which is a threading intercalator, so it intercalates at GC-rich sequences, and then it has cyclic peptides that wrap into the minor groove. And when DNA is pre-exposed to that molecule, we see very little damage by this compound here. And so it appears if you have, say, a minor groove binder or intercalator alone pre-exposed to DNA, this is still active, but with actinomycin D, we see nearly total inhibition. So it's behaving very much like a threading DNA intercalator. In terms of other advancements in click chemistry, we've had a recent work in nucleic acids research led by Simon Poole, there's these groups of compounds called clip phens, where a serenol bridge clips two groups together to maintain this 2-to-1 stoichiometry. We wondered, if you use click chemistry, could you force two of these phenanthroline groups to bind with two copper ions, and therefore to develop an enhancement towards DNA damaging activity. And I won't go into a lot of detail about this, but we've crystallized the structure. It's an extremely active DNA damaging agent, which is copper dependent. And we ran a lot of functional assays and engaged with the NCI 60 to characterize its binding properties. And this is recent research in NAR. And off topic slightly from AMNs is our interest in trinuclear platinum compounds. And we've also found a way to introduce a asymmetric azide into this triplatin NC scaffold. This binds electrostatically to the DNA backbone. But the advantages of having this azide in the future is obviously for directing groups. But in this study, what we were able to do is look at the applications for in-cell click chemistry. And we could find that it was nucleolar targeting within triple-negative breast cancer cells. And this work was also published last year in Nucleic Acids Research, and is the basis now for future work in terms of click chemistry-based targeting. So in the short time that I have remaining, I'll just talk a little bit about these catalytic oligonucleotides. As I mentioned, Dr. McGorman will speak in more detail about this. I mentioned earlier about our collaboration with Tom in these European networks. The idea behind this is really simple. And this is, can we use a nucleic acid click chemistry to attach a label? And this group here could direct a gene targeting group. For example, a triplex forming oligonucleotide can be used to direct a cleaving system. So this TFO then could be functionalized with a AMN or some other group like a platinum group. The idea is that you have sequence-specific interactions, and then this cleavage activity can occur as a result of the localization under the influence of the TFO. And we've obviously have an interest, and we've published standalone compounds in terms of their DNA damaging properties. These are copper-based and platinum-based, and we've then successfully made stable azide derivatives of these families and published them when we've under the attachment of TFOs for these kind of gene-targeted applications. So I'm not going to go into a lot of detail about this, but I just want to highlight maybe two areas that were both led by Brina that I think are really maybe interesting for future development. One is on the use of single nucleotide incorporation primer extension. So in this case, we've been able to click on a copper chelating DNTP at a single site, and then using primer extension, complete the synthesis of the TFO using this template, and then perform and then separate both of them, and using this kind of enzymatic TFO for various applications. And what Brina did then was to construct a GFP plasmid, and we were able to find that in a challenge assay with PUC19, only the GFP plasmid was cleaved, and we could, using Sanger sequencing, then we were able to identify that if you have no modification, you get cleavage at the triplex duplex junction. If you have a intercalator, you can cleave closer to the modification site. And then if you have a DPPZ group, which again improves intercalation further, we can get cleavage very close to the modification site. And so all this work was done in collaboration with Tom and AFAF, and I think has a lot of future potential for other modifications. And very lastly, one of the things we're interested now in is developing ASOs, and these would be functionalized with artificial ribonucleases, so ARNs. And so the state of the art then is ASOs and obviously ribotacs in terms of their applications. But our approach here was to functionalize an ASO with this artificial ribonuclease. And so Brina developed a range, or we looked at a range of copper compounds that are available in the group, and targeting the stem loop of the SARS-CoV-2 viral genome. we were able to identify this particular ASO5, which had the highest activity or affinity, and we functionalized that then with locked nucleic acids. And this work now is at revision stage in Angevin Chemie. We just have a functional assay to run. So this is quite exciting as a new method for directing metallo drugs to RNA effectively. We have some high throughput screening facilities in DCU. One of them is this autopilot system that can we interface cell imaging along with cell screening, and this can be done in large arrays. So it's not just cytotoxicity, but we can run multiple plates and perform kind of complex endpoints, including flow cytometry. So this system is very close to being finalized. A PhD student in the lab has been kind of instrumental in pushing some of this work, and we're hopeful we'll have some data to show future meetings related to this autopilot technology. And finally, we've been successful with this recent funding for a C-trap single molecule imaging. So if anyone is interested in collaborating in either of those areas in terms of the autopilot or C-trap, please just ask. We're really interested in collaborating with the community. And finally, thank you all for your attention, to Tom, obviously, and to all of our collaborators. Thank you very much.
Any questions?
That was a very nice talk. I have a very technical question.
Mm-hmm.
So you produce a lot of these ligands with kappa-1 click chemistry,
Mm-hmm.
and then you exchange them with kappa-2, or is there any doubt that maybe some of the compounds do still contain kappa-1?
Yeah, that's a great question. So we obviously characterize all of our hybrid systems to ensure that there's no copper remaining in the after the click reaction. So you can use a number of reagents like EDTA wash is generally acceptable. It generally works. There are other, if some of the ligands are difficult in terms of releasing copper, you can use other types of ligands, but we will validate and verify that there's no latent copper there left over after the reactions. And we see that because when we run the hybrid without any metal ions, so the APO system, we see no damage, or at least we can't detect any damage. So if there is copper in there, it's at an extremely low level that doesn't affect any of the interactions. The second thing to say is that when copper is in there, or we have other metal ions like platinum, for example, they can often be electrostatic. So you see an enhancement for the, in terms of the association kinetics for the hybrid itself, because you've got both the kind of non-covalent element and then the group itself, when it's cationic, you generally get an enhancement that can be detected by, it's very small, maybe 2 or 3 degrees in terms of stabilization in UV melting or thermal melting, fret melting, for example. Okay.
I have a follow-up question. Do these systems and complexes work with kappa-1, or are they only working with kappa-2?
So they require copper-1 to work because basically you need to, so from a technical point of view, so copper-2 is D9, copper-1 is D10, and that's the molecule that interacts with dioxygen. So what happens then is once it binds with dioxygen, you get an electron transfer from copper-1 onto what you get is a superoxo species, and that's the agent that damages DNA itself. It's not a free, like Fenton type chemistry, so it's slightly different. So if you've copper-1 in there, it's immediately active, but generally we add copper-2 and then add like ascorbate, or if it's in a cellular environment, it's producing, there are systems that are self-activating. And are you speaking, Breen? So there's a talk later from my colleague that will speak about self-activating systems, and that might help answer that question a bit further.
Yeah, thank you. That would have been my last question. If you could use just one or the other species and then redox.
Yes, so self-activating systems.
I will wait for
the electron transfer. Yeah. Okay.
Thank you.
One final short That's very nice talk. For the ASO-guided metallo-nuclease, how do you separate the effect from cleavage induced by RNase H1?
So it's really quite interesting because that study, which is under revision, first of all, the gap is quite small, and we were surprised to see that with that system, not only when you have a chemical modification like the alkyne group, you can still recruit RNase H, but when we click on a cleaving group like phenanthroline or something else, you still get RNase H activity. So we're seeing a dual type of mode of activity, which is quite interesting because quite often that's some of the reasons why you see incomplete activity from ASOs. And so we're hoping to see a kind of latent type response within that system, but we can see both, and RNase H seems to be happy enough with a bulky group at that site. Yeah.
Thank you.
Okay. Okay.